Onwards to one of the most studied machineries in biology, protein synthesis, where all of biological function lies. This is given true by our genetic code, which is encapsulated in DNA. First, let's clarify a certain set of points. The first thing I want to talk about is a gene. How exactly would we describe what a gene is? I'm going to try a thinner pen. Nope, that's not good. Yeah, that's good. A gene is defined as a segment of DNA. Let's put some context into that. Now, DNA, we know it's a long molecule. DNA is a larger molecule, rather large molecule. If I consider just this part, that can be called a gene. That can be called a gene, simply a part or a segment of DNA that codes for a particular protein. That's the most important part. My handwriting is screwed. Particular protein. So this gene should produce a certain protein. I'll call this protein A. Right. Back to the definition. A gene is a segment of DNA that codes for a particular protein. This is a very simple way to put it. Now I'm going to give you a formal definition as to what a gene is, because this is going to be important when answering questions. If I gave you the definition first, you might not have understood completely. I just wanted to give you the context first. Okay, let's go. A gene is a sequence of nucleotide bases, meaning simply a small segment. Sequence of nucleotide bases that codes for the production of a specific sequence of amino acids. So a sequence of nucleotide bases that produce a specific sequence of amino acids. Okay, if that is getting too much, you'll understand this entirely once this chapter is done. So do hold on to your horses. Do take down the definition in any form of way. Do follow along with this series as much as you can. Just try to pay attention, and you will understand all the concepts I'm breaking down for you. Production of a specific sequence of amino acids, which in turn make up a specific protein. We have to move to a new line. It's protein. Okay, never mind. So let's see. Just some more context. We have a sequence of bases like this. Those bases are going to produce a specific sequence of amino acids like so. And those amino acids, later are going to fold into some form of a specific protein. Some kind of a protein. It's going to form. Simply. That would be the formal definition of what a gene is. That would be the entirely laid out definition. Most of the time, this is a 3-mark question. Or sometimes a 2-mark question. But most of the time, it's a 3-mark question. In the world of a gene, we have something called a codon. Now I'm going to write down a specific point. Each codon, C-O-D-O-N. A codon is a triplet of bases. Codon is a triplet of bases that code for a particular amino acid. What would that look like? Okay. Now, when you're reading through your genetic code, of course, you have bases. Even previously, I represented our bases like this. Now, we know, obviously, we have A, C, G, T, A, G, C, T, T, A, C, G, A. Like so. Every single time DNA or a gene is interpreted, we are reading 3 by 3. We're going to be reading 3 by 3. So ACG is read first, TAG is read then, CTT, ACG, and so far. And so far. I'll draw another line just for clear distinction. Do you see? We're reading triplets of bases. Every single time a triplet is read, we call it that a codon is read. A codon is simply a triplet of bases. Each codon is going to be able to code for one specific amino acid, which is the building block of a protein. So ACG might code for this. TAG would code for this amino acid. And then CTT would code for this particular amino acid. ACG, now you see, ACG here, we have another ACG. So this is also going to code for the exact same amino acid that ACG codes. That ACG codes for. Now this is known as the universal genetic code. This is known as the universal genetic code. I'll describe some features of the genetic code so that you're going to understand why this is so good and why this is so important. The first feature, there are 3 features. The first thing you should know is the genetic code is universal, meaning it is the same throughout all the organisms you're going to find in the universe. Meaning every single organism is going to be coded for by ACG, TAG, CTT, ACG, A, T, C, G, those stuff. Why? Because we evolved from one central organism. We evolved from a universal ancestor. That's what science believes. That's what science has brought to prove. All organisms share the same universal genetic code. The second feature is that the genetic code is degenerate. What does that mean? Meaning, if there's a problem, a small misfire, it can be accounted for. That means multiple codons can code for the same amino acid. For example, now we just said that this ACG codon, so ACG codon would code for this amino acid, this particular amino acid. Due to the degenerate nature of the genetic code, there might be 2 or 3 more codons that code for the same exact amino acid. Maybe CGG also codes for it. Maybe TGT also codes for the same exact amino acid. Now, why is this there? This is there because even when our DNA mutates, we can still try to preserve as much function as possible. Now, what on earth is DNA mutation? That's the last part of this chapter, so I will not be diving deep into that, and you don't have to understand too much context when it comes to that. Just so you know, the genetic code is degenerate. The third kind of function or feature that the genetic code comprises of is triplets or codons. That's the third feature of the genetic code. We have codons, including start and stop codons. Okay, now let's just take a small step back. So here we have talked about codons and triplets and amino acids and the genetic code. This is also confusing until I'm going to bring in a specific diagram / table. After looking at that, you will understand how the genetic code works and how it is linked to each and every organism. And this would be the said diagram / table. This is simply a complete representation of all the known codons to mankind. I wouldn't call it all the known codons, but basically this is going to give you a very good enough throughput. Now, do you see, every single time we just talked about how a codon is always going to be made up of 3 letters. That's why all these are indeed made out of 3 letters. See, all of them, they're made out of 3 letters. That is what you call a codon. And each specific codon is going to code for an amino acid. Now, in biological reality, we know that we have 20 different types of amino acids. Only these 20 different types of amino acids come together in different manners to produce the ultimate throughput of a protein that we can biologically observe. Let's just take a look at with 1 or 2 of these things. Now, if you have a AUC codon, that's going to code for this specific amino acid. Now, what is that specific amino acid? Each and every amino acid has a specific name. LEU is for leucine, and then we have serine, proline, alanine, valine, methionine, arginine, glycine, different types of amino acid. If you want to know the structure of an amino acid, do go back to biological molecules. Under that, the structure and the 20 different types of amino acids are explained in a lot more detail, in a lot more context, so that you can just couple things together from this and that chapter together to understand things better. Now, do you see, we just talked about how the genetic code was first universal, obviously, and then we talked about the degeneracy of the genetic code. That would look something like this. Now, do you see? Let's take proline, PRO. PRO stands for proline, okay? This is an amino acid. You don't have to at any point memorize all the amino acids. You don't have to memorize the codons, okay? Don't be stupid. Don't try memorizing all the codons. If you want to just improve your answers in the essay format, you can just remember 1 or 2. Maybe you just remember that so you just remember that CCU codes for proline, something like that, just 1 or 2 bits you remember, and that would give you an upper hand when it comes to answering and pleasing your dear examiner. That's just a tip I would like to give, but it's not necessary at all. Now see, the amino acid proline is coded for by 1, 2, 3, 4 different codons. If you have a CCU, it's going to be proline. If you have a CCC, it's going to be a proline. If you have a CCA, it's going to be a proline. If you have a CCG, still you would be coding for the amino acid proline. That explains the degeneracy of the universal genetic code. Multiple codons are coding for the same exact amino acid. And then finally, obviously we know now the genetic code is made out of triplets or codons, and that includes start and stop codons. Why do we need these start and stop codons? Now, when it comes to protein synthesis, first of all, we have to begin synthesis, and we should also have a point where we can end that synthesis. We have to always have a point to end, otherwise it's just going to completely transcribe the entire straight. Now don't worry about transcribing and producing amino acids and proteins, that will be discussed in a bit. Here all you have to know is, see, there are specific stop codons listed in this table slash diagram. One example would be UGA. The moment that this UGA codon is met by something, that something I'll talk about in a moment, but once this stop codon is met by this something, protein synthesis stops. Protein synthesis stops. Okay, protein synthesis is going to stop. Now I see two more stop codons. UAA also seems to be a stop codon. I'll highlight those in red. UAA, UAG, and UGA seem to be stop codons, and these are the three stop codons that are in your genetic code. Now if you see, they're almost the same. UGA is UAG, UAA. They're very similar, and all of them begin with the base U. Okay, why do we have U? Do you remember that U was part of RNA? Okay, but then we're talking about DNA. Why exactly do we have U here? Don't worry about that. That'll become obviously clear in the next spread once we get to transcription and translation and where proteins are produced. This is the nature of the genetic code, and we have three specific stop codons like this. We have three specific stop codons like this, and this is the entirely universal genetic code. Right, now let us focus on the process of protein synthesis entirely. So I'll just call it the process underline. Protein synthesis is a 2/3 step process. Why do I say 2/3? Let's see. First, you have a process known as transcription. Now, transcription, what it does is, we know all the information we need to produce a protein is encoded in our DNA. DNA is a very lengthy molecule that is stored inside the very small nucleus. And whenever we are producing a protein, whenever we're producing a sequence of amino acids, we don't need the entire thing. We don't need information from this entire thing. I want just a small part of it. Or else, in more biological terms now, I just want a gene. Alright, I simply want a gene. Now, if I just want a very little part of it, this is inside the nucleus now. Should I take the entire huge DNA molecule out of the nucleus? No, that's not required. It's not very efficient. So I just need this little part. So from DNA, I'm just going to copy that gene, the only part of the DNA that I want, the only gene, onto a small RNA molecule. And then I'm going to take it outside the nucleus into the cytoplasm, where I can translate it. What happens in translation? Now I have the RNA molecule. This RNA molecule can be utilized to produce my overall protein that I need. That is the overall process of protein synthesis. Now, why do I have a small space in the middle, and in the beginning, what did I say? 2/3 step process. That's because at AS levels, you should know that there's something known as post-transcriptional modifications. and then only translation takes place. Now in IGCSE, it was a straightforward two-step process. Transcription number 1, translation number 2. Still, that's the idea. Those are the two main things, but there's a small sub part. We'll call it 1.5. This is known as post-transcriptional modifications, and we will be looking at what these post-transcriptional modifications are in a little bit more detail. Let us begin with transcription. Number 1, or step 1, transcription. Let's see what transcription is. Transcription can be described as the process of transcribing. Transcribing is another term for copying. Or simply copying DNA onto an mRNA molecule. Okay, now here I said DNA to RNA. Now I'm just going to edit that and say mRNA. What is this mRNA? mRNA is a very specific type of RNA you find in the process of protein synthesis alone. This is known as messenger RNA. Simple M, capital RNA. Messenger ribonucleic acid. Why do you call that? Why do we call that? That's because it's almost taking a message. The gene is being taken as a message into the nucleus, by which the cytoplasm can then produce the protein using that to mRNA molecule, and this occurs in the nucleus. Okay, this occurs inside the nucleus. Did I spell nucleus wrong? Like so. Let's just look at this process in a little bit more detail. Okay, I'm going to draw some DNA. Okay, that is some DNA, and we know we have obviously bases on that DNA. Now let's say this is the length of DNA I want. I just want this bit, just this part. So from this strand, I'm taking things. So I'm going to call this the template strand. I'll call this the template strand. This is the template strand. We transcribed strand, something like that. Now we have to produce a small segment like this of RNA, or in other terms of mRNA. This is being done by a very specific enzyme known as RNA polymerase. Now don't mix this up with DNA polymerase or primase or anything. This is a completely separate enzyme only involved in protein synthesis, not DNA replication. Those are two distinct processes. They are very close by in these chapters, so it's just viable that you get it mixed. So do not do that. RNA polymerase is just the enzyme that you need to produce a small strand of messenger RNA containing a gene, containing a sequence of the amino acids that are going to produce a polypeptide or a protein. A polypeptide or a protein. Here it's just this mRNA strand. Now I have my mRNA strand. So that was the ultimate goal. So let's look at what exactly happened here in a little bit more detail. Let me type things out. So part of DNA unwinds. That's the first thing. We need to get information, and I told you we can't do it when it's in its double helix form. For us to get information from DNA, we have to unwind it. Now here you don't have to mention what enzymes are involved in the unwinding process, nothing like that. So free activated RNA molecules, or in more specific terms, RNA nucleotides, pair up via hydrogen bonds. What does that mean? I didn't talk about that at all. With the complementary exposed DNA. All that ruckus is just talking about how RNA pairs up with this DNA in a complementary manner. Now let's look at this complementary manner in a little bit more detail. My grammar is just going way off today, I swear to God. Okay, if I have a TGCATCG. Let's look at what's going to be found on the mRNA transcript. Right, now we know adenine will always bind with thymine and guanine will always bind with cytosine. But we also learned in RNA we don't have thymine, but we have something known as uracil. We have something known as uracil. So because of that, here I can't say T. On the mRNA transcript, I can't possibly have T. That's wrong because RNA doesn't have the thymine base. Instead of thymine, what RNA has is uracil. Now thymine pairs up with adenine, that's fine. RNA has adenine. So guanine, cytosine, cytosine, guanine. Again, adenine pairs with uracil, thymine pairs with adenine, guanine, and cytosine. So that's the key importance of what you should know, and that is why codons contain the U base. Because codons, protein synthesis coding codons, are RNA based, and RNA does not have the T base. RNA only has the U base, not thymine but uracil. Right, so complementary exposed DNA is replicated like so. Now then comes the enzyme DNA, not polymerase, not RNA. It's RNA polymerase. Wait, what? Hold on, what did I say? My lord. I said DNA, not RNA, then not polymerase. Okay, let's forget the last 5 seconds ever happened. The enzyme RNA polymerase. I am so overworked today, that's why. Sorry about that. The enzyme RNA polymerase bonds the sugar phosphate backbone of the mRNA strand. This is just saying that the enzyme RNA polymerase is what actually produces the RNA molecule finally. Bonds the mRNA strand. Now after transcription or this transcribing or this copying state is done, the hydrogen bonds reform between mRNA and DNA. Not reform, hold on. It's between, not reform, the H bonds between mRNA and DNA. There's no reforming required. So here, look at my mistake. These things are just bound by hydrogen bonds. Now, for me to actually remove this mRNA transcript and take it to a separate place, I have to break those H bonds between the molecules. mRNA and DNA break, and thus the mRNA molecule is removed and DNA can recoil. After that's gone, DNA will go back to its recoiled form. Then now I have a nice separate mRNA molecule. This mRNA molecule then leaves the nucleus via nuclear pores. That's all. Simply via the nuclear pores, this RNA molecule is going to move out of the nucleus. Because now we have to get it into the cytoplasm, because translation happens in the cytoplasm. We'll talk about that like this. Transcription occurs in the nucleus. Translation occurs in the cytoplasm. So we should have some form of method of getting the mRNA transcript from the nucleus all the way into the cytoplasm. Okay, alright, now this little tiny little mRNA molecule is outside the nucleus. Now it's outside the nucleus and it's going to undergo these post-transcriptional modifications I was talking about. Post-transcriptional modifications. I will not be talking about these post-transcriptional modifications right now. I'm going to focus on translation and then we can go on to that. Okay, so transcription done. DNA, now I have a working mRNA template. we're going to do some post-transcriptional modifications onto it, and then we're going to finally translate that. Now, after these post-transcriptional modifications, the mRNA molecule is now in the nucleus, and it can undergo a beautiful translation process to produce a protein we need. Now, let's type things out like I did before. This is the process of translating the mRNA molecule into a polypeptide. And like we said that transcription occurs in the nucleus, this guy occurs in the cytoplasm. So we needed some form of manner to get that genetic information from the nucleus all the way onto the cytoplasm. That's why we needed an mRNA template. Now this is a very simple process. Even the previous one was very simple. Now let me take the mRNA molecule and display it like so. I think I'm going to need a little bit more space on top. Right. So let's write down the bases by aligning things to my grid. I'll try to have multiples of 3, right? Okay, let's actually use actual codons. What's my this thing? Okay. CCU ACU. CCU ACU. CCU ACU. Let's move that a little bit here. Let's find another codon. GCU GGU. GCU GGU. GCU GGU. Okay. Now let's say the mRNA transcript that came to the nucleus, the cytoplasm, is this. It's composed of this sequence of bases. This is the mRNA transcript. Now let's look at what happens. Translation occurs in the cytoplasm. Yes, very true. But translation cannot occur without this guy. This is a very well developed ribosome. Ribosomes have a large subunit and they have a small subunit. You don't have to know about that much, but the idea behind the large and small subunits are that, let me see if I can select this in one go. Yes, the large subunit binds like this and the small subunit is going to bind like this. That's going to allow the ribosome to scan this mRNA template. So I'll try to select it again. Can I select both? Nope. So this is just going to read the mRNA template, and then it's going to translate it. So just to be clear, happens in the cytoplasm and requires a special cellular organelle known as a ribosome. Right. Now this ribosome is going to read this. So when this ribosome reads this, it's obviously going to scan for the first codon. So it's going to read the first triplet. It's GGU. So universally, it is known that GGU, let me highlight that right here. GGU is right here and it codes for the amino acid. GLY means glycine, and that's the simplest amino acid you're going to have. GGU codes for glycine. So glycine will be added onto a growing polypeptide chain. But where's glycine? Where does glycine come from? Does it come with a snap of a finger of the ribosome? No, ribosomes don't have fingers and they clearly cannot snap. Okay. The thing is, if you have been eating enough proteins, if you have been taking enough proteins as your daily balanced diet, chances are you have some very good healthy proteins, therefore absorbed amino acids already circulating in your blood and therefore in your cells like this. So rather proteins, not proteins, amino acids. But the thing is, these guys can come and walk onto this all of a sudden. They can't just come and join. For that, you need a special kind of molecule carrier known as a transport RNA. You call it tRNA for short. The T stands for transport. We need a transport RNA. Okay, now we know GGU will code for glycine. Let's say the orange one is glycine. So what's going to happen is this tRNA molecule is going to pick up glycine. I'll bring glycine forward. Nice. And then he is going to go and connect onto the GGU. Let's make it a little big. So GGU like this, and boom, the first amino acid has been successfully added. The first amino acid has been successfully added. Right? Then that's fine. Those three codons are done. Not those three codons, those three bases, that one codon is done. Then we're moving on to what? GCU. Let's just go to our diagram and see what GCU is going to code for. Okay, right here. GCU is right here, and we're coding for alanine, yet another amino acid. Let's see. Okay, let's say the pink one is alanine. So again, another transport molecule or a transfer RNA, tRNA, is going to carry this molecule like so. It's going to pair up with the codon. You see, that's why it has 3 legs. Okay, should make it larger more. See, the 3 legs are going to pair up with the 3 codons, and then this is added, and a bond is going to form in between these 2 amino acids. And we clearly know if we have a growing chain of amino acids, we get a polypeptide, which can then form a protein, right? And then the same exact thing occurs again. Let's see what ACU would code for. You don't have to memorize this table at all. I'll repeat that. Now ACU is going to code for THR, which is known as threonine. Let's say that's the green one. Again, I need a transfer RNA molecule. Without that, the amino acids are not capable of moving. So he's going to come and bind like this. Let's make it large. Bind like this. Again, the third polypeptide, the amino acid has been added to the polypeptide chain, and boom, after some time, we're going to have a growing chain of amino acids added like this. So this one, then the pink guy, then we'll have the green guy, maybe the next one is the blue one, maybe again we have an orange guy, maybe again a pink guy, maybe we have 2 blue guys then, not just one. Whoops. Like so, and we're going to have a growing chain of polypeptides, growing chain of amino acids, which can produce a polypeptide chain, and that is going to fold into a 3D structure, which is known as a protein. Now that was a lot of information, but I wanted to outline that as a diagram. Let's type out what exactly is happening. Now the first thing is the mRNA strand attaches to the ribosome. The mRNA strand attaches to the ribosome. tRNA, transfer RNA. tRNA for short. tRNA. Molecules with amino acids. So they're bringing amino acids. These tRNA molecules with amino acids containing, okay, listen to this closely, anticodons. Anticodons. If this was a codon, meaning if GGU was my codon, tRNA molecules have anticodons, meaning what's the opposite of this? CC, uracil would bind with adenine. This is my codon, and this is my anticodon. Okay, that's the only way in which tRNA molecules function. That's the only way that they can bring complementary amino acids. Because they have anticodons, they can be complementary to the codon. Thus, we have anticodons, and therefore, because of that, we can safely produce a growing polypeptide chain. They have anticodons. They are free in the cytoplasm, like I mentioned. Okay, the complementary tRNA molecules are bound to the mRNA strand by the ribosome. Without the production machinery, without the ribosome, none of this is possible. Okay, that's why ribosomes, even whilst being nanometers in size, are very important. And then each polypeptide that the tRNA molecule, there's a mistake in this. I'm going to correct that after I'm done with this. Each polypeptide that the, let's say, tRNA molecule carries is successfully bonded to each other via, what are the bonds that form between amino acids? That would be peptide bonds. And like we said, after the stop codon has been met, tRNA will not bring any more amino acids, okay, at the stop codon. What are the stop codons? Let's see. The stop codons are UGA, UAA, and UAG. After these stop codons have been met, guys, we are not going to be bringing any more amino acids into this, sorry. Simply speaking, we're going to stop the process of translation. After each stop codon initiates the end, the newly formed polypeptide chain folds into its specific protein. Right? Now, what's the mistake I told you that was in this? Does each tRNA bring a polypeptide? No. Each tRNA brings an amino acid. I just want you to be clear on that distinction. I also kind of messed up the words, I think, a couple of times. One amino acid, if you have a growing chain of amino acids, that's known as a polypeptide, which can then form a 3D protein. Okay, small distinction, but clear marks can be lost if you mess things up. And those are the two overall processes of transcription and translation. Now let's look at what post-transcriptional modifications looks like and what they are. It's not complex. It's not a completely separate process. You just have to understand what is kept and what is removed, and that's all. Let's see. Okay, so now once the mRNA molecule has been produced, now forget about translation for the moment. We're here, okay? We're in the middle. After transcription, we're making the post-transcriptional modifications. After all of the transcriptional steps are done, we have this mRNA template. Okay, we call this the primary transcript. We call this the primary transcript. Now the problem with the primary transcript is, let me use a smaller pen size. The primary transcript contains both introns and exons. So I'll write it like this, both exons and introns. These are going to be two new words for you under genetics. So let's clarify what each of those things do and how they function. Exons code for amino acids. Exons code for amino acids. Introns do not code for any amino acid. So now if you say for amino acids ... if you say out of these two, what are the important ones? Do we need exons or do we need introns? We clearly need the exons. We don't want the introns. Now DNA is a very complex and very confusing guy. Okay, he has spaces, he has hyphens, he has dashes. So those things are also expressed as introns. We call those non-coding regions. And so many polymorphic studies have been done during genetic research under these introns and exons and polymorphic non-coding regions that can cause different types of diseases. It's a completely vast type of study, completely relevant to you as well under this level. All you have to know is exons code for the amino acids, but introns actually have no use because they don't code for any type of amino acid. Now, we don't want garbage. No, we don't really need garbage. So post-transcriptional modifications ensure that the introns are removed and exons are fused together. I hope you can understand my handwriting. This is just dying at the moment. You take away the introns. We don't want introns. They have to be removed and the exons are fused together. And after that is done, I get a mature mRNA molecule. So we started with the primary transcript. Now I have a mature transcript. Primary transcript, mature transcript. What would that look like? Let's just have a small go at it now. I'll indicate a couple of regions. 1, 2, 3, 4. Purple. Okay, purple represents exons. Do we need exons or do we not need exons? We need the exons. The blue regions would show the introns. Do we need the introns? We don't. We don't need introns in any form of manner. I will also indicate that exon, intron. This is the primary transcript. Now I want to produce the mature transcript from this. All I have to do is now I'm going to replicate this, duplicate this, like this. Now I don't want any intron. I'll get rid of the introns. I'll get rid of the blue ones. No need. Not coding for anything, so it's useless to have. And then I will fuse the exons together like this, thus giving me my final mature mRNA transcript with only exons. And those are the post-transcriptional modifications that you need to be aware of. It's not hard. Just to recap the entire process and all of the relevant theory. The genetic code is a universal code that codes for specific amino acid, hence a polypeptide and hence a protein. Here you can have a go at all of the codons that make up the universal genetic code. And then you know the process of protein synthesis is a 2/3 step process where we have transcription, post-transcriptional modifications and translation, helping us produce the overall polypeptide chain or the protein that we desire.